Hello, I'm Wayne Brough, AHDB Knowledge Exchange Manager, and welcome to this AHDB webinar examining the selection and use of biological control agents in ornamental crop production. In this, the second of two webinars, we focus on cell-piercing pests, primarily mites and thrips. Before I introduce the two speakers and their presentations, just a few housekeeping points to address. You'll notice the audience is in mute. But please don't let that stop you from posing questions, and I'll come back to how to pose a question in a moment. In terms of timings, the webinar will take around about an hour, give or take 5–10 minutes, depending on the number of questions we have. It will be recorded, so if you need to leave or if you need to pass it on to colleagues, it will be available on the AHDB website in about a week's time. There is an handout available containing all the slides from today's presentations, and I'll come on to how to download that in a moment. And you should have been sent basis forms for this at registration, so that should be all taken care of. If not, please contact my architect after the event, and we'll sort that out. In terms of questions and handout, you should see something like the right-hand side there, the menu on the right-hand side on your screen. Before you click the red button with the white arrow to downsize that particular menu bar, to draw your attention to the handout and questions. If you click the handout grey bar, the drop-down menu should highlight the handout and also the basis form. If you want to download the handout, please just click the handout and download it during today's presentation. And again, in terms of questions, if you wish to pose a question, please click the grey bar, and then from the drop-down menu, type in the question. Please type in questions throughout today's webinar. I'll keep an eye on them and pose them to today's speakers. And again, the recording will be made available next week on the AHDB website. In terms of today's webinar program, we've got two speakers. Neil Helyer will be talking about the mechanics of mite and thrips management, and James Lisher from Cobbins Nursery will be talking about the management of thrips setosus at the nursery. Just to say a few quick things about each presenter. Neil is well known to the industry, having been the IPM specialist at Fargrow since 1995. And before that, he's undertaken 20 years of entomology research at the Glasshouse Crops Research Institute. Unfortunately for the industry, Neil has now recently retired from his role. He is the director of Cobbins Nursery near Worthing, West Sussex. The nursery produces a wide range of bedding and pot plants for garden centre sales. So if I can ask James to close his webcam and mute his microphone, we will commence with the webinar and Neil's presentation. Thank you.
Good afternoon and welcome to part 2 of the use of biocontrol agents for various pests in ornamental crops. Before we start, I just want to introduce you to the Syngenta Ornamentals podcast. I must admit, I only came across it the other week and I think it's actually very, very useful. So it's worth making a note of the Syngenta Ornamentals podcast. It's very useful. Also on podcasts and webinars, Fargrow have been doing a few short podcasts, usually coming out on a Thursday morning during lockdown. Again, these are all available online. It's worth contacting them just to get the latest, because they're very short, sharp, 5 to 7 minute podcasts. Okay, so this is going to be looking at the various species of thrips, species of spider mites, looking at the biological or biology of the pest, the controlled biological and chemical and IPM compatible products. So to start with, we're going to look at the two-spotted spider mite. This is Tetranicus urticae, also known as the red spider mite, but more commonly, I say the two-spotted spider mite, TSSM. The damage they do is a very fine speckling visible from the upper surface of the leaf. So the top side of the leaf, you'll see the damage. Turn the leaf upside down and you should start to see a few absolutely minute spider mites. Okay, the life cycle. The two-spotted spider mite has males and females. Females have a more rounded rear end and they lay perfectly spherical eggs. The eggs can be laid on the leaf surface or between layers of webbing that the mites produce. So they're not always going to be on contact with the leaf. Those eggs hatch to a six-legged larva. Now this makes them spider mites and not spiders. A true spider has 8 legs right from the egg hatch. It then goes through 2 resting stages where another pair of legs are sprouted, and we have the proto-nymph and the deutonymph, and that molts each time and sheds through to a male or female mite. If you look at the temperatures and the days it takes, the higher the temperature, the more rapid the development of the spider mite. And again, once they've reached adulthood, we're anything between half a day, 12 hours, before they can start laying eggs. So it's a very, very rapid reproduction cycle. The term red spider mite really refers to the orange-red diapause phase. Now that is a naturally occurring event, 2 out of 3 naturally occurring events. So reduced day length, reduced temperatures, and reduced plant nutrition. If the mites receive 2 out of either of those 3 events, they can go into diapause. So if you're growing a crop under heat in the winter, but under natural day length, and you're keeping the crop nutrition good, you can have spider mites all year round. It needs 2 triggers to make them go into diapause. Diapause can be in the structure of the greenhouse. The top photograph there shows a support bar, a stanchion, and you can see the spider mites accumulating around the top of the stanchion. Now those go inside that pole. Absolutely, you have no way of controlling them over winter. You're never going to get a spray down inside them. The eye protectors, the cane tops, brilliant for spider mites. They will accumulate inside bamboo canes and under those eye protectors. Again, a lovely winter job. Whip them all off, sterilize them in boiling hot water, pop them back on again. That will also help control any mealybug that may be in that greenhouse. The most favored position is under the rim of a pot. So if you've got your plastic pots and with a rim on them, they will overwinter in underneath that. You need to turn the pot upside down and you'll see the spider mites accumulating in groups of maybe half a dozen or 20 at a time into those little red patches of red spider mites. Again, there's no spray is going to work through a plastic pot and go up and under to kill them. So it's quite critical to aim for spider mite control before the mid-October cutoff when they start going into their winter diapause. So that was a little bit on the Tetranicus, the common spider mite. Just want to introduce you now to another spider mite. This is the Eotetranicus, the Lewis mite. Please note in the bottom there, this is a notifiable quarantine pest. Up until the 1st of January this year, it was a regulated pest. It's now, because of Brexit, become a quarantine pest. So it's not easily controlled by Phytosulus. We have to look at other ways of controlling this particular spider mite. The damage they do is very similar. There's a range of crops that we know it attacks, and that list is probably going to grow over the next few years. So it is a serious pest. It is one to take notice of and just be prepared to find it. So really you need to be looking at certain crops. So poinsettia, it has definitely been found on in the UK. Just a few basic details on this one. So it's slightly smaller than the two-spotted spider mite. It doesn't have the 2 dark spots, but has smaller dark spots. So not as big as the two-spotted spider mite. The rest of it is pretty much the same. But notice where it comes to the biologicals, Phytosulus is not that good. So we're looking at Andersoni or Californicus as giving better control against this particular pest. Chemical-wise, there's not a lot of resistance at the moment. So thank goodness we can still use a few pesticides if we need to. And because it's a notifiable pest, you will need to go for eradication before you're allowed to move those plants. Okay, that was the Eototranicus genus. Now we're looking at another genus of spider mites. This is the Panonicus. These can be Panonicus citri, the citrus spider mite, and Panonicus olmi, which is the fruit tree spider mite. More or less disregard that one for this presentation, but the citrus spider mite is most definitely a pest we're finding on a whole range of plants with a similar leaf structure to citrus. So the photograph shows Skimmia. In this instance, the grower phones up, says, we have spider mites, introduce Phytosulus, doesn't like Panonicus, so the damage got worse and worse. Again, it is critical to identify your spider mites and identify, if not, just get one of your advisors out, get someone to identify what the spider mite looks like. They do look different. They're that darker red and much hairier over the back. So it's not impossible to identify them. The other point on Panonicus species, they overwinter on the plant itself. So you get these small orangey red eggs laid on the plant, not necessarily off the plant. So it'll be evergreen plants. So again, Skimmia, Choicea, that sort of plant where we're finding this particular spider mite. Another genus, the Oligonicus. Now this is another specific citrus conifer plant. That's the conifer spinning mite is the common name for it. It spends its entire life on that particular host plant. It is becoming a nuisance on ornamentals, particularly like cupressus, which can come, or the spider mite can find its way into it. Don't forget, although spider mites don't fly, they can float on air currents. And so they can find their way into a perfectly clean house and just make a nuisance of themselves on a completely clean crop, just out of the blue, basically. Again, different mites, we're looking at the broad and gall mites. This would also include things like tarsonemid mites. These are minute. We're talking about a minimum of a 20 times hand lens to see anything at all. So your standard 8-10 times hand lens is not going to pick up these mites. However, the damage is quite distinct. It's usually a distortion, a twisting of the leaf veins, but a shiny on the underside. This is the broad mites. The term broad mite doesn't refer to the size of the mite, just that it's a polyphagous, look at the name, Polyphagotarsonemus, fantastic name. So poly, many, phago, eat, and it's a tarsonemid mite. So it's a very broad host range of plants it can damage. The Aculops is the fuchsia gall mite. So some mites, as they feed, their saliva is toxic to the plant, causes a growth, a distortion, a gall or whatever, and the mites lay their eggs inside that gall. So they're not laid on the surface, they're laid within the gall where they're protected. So again, a lot of your contact sprays are not going to have the effect on this particular sort of mites. Okay, that was looking at just some of the spider mites. There are more. Looking at the control with the predators, I started off with Phytosulus, purely because this is the main one that if you phone a supplier and say, I have spider mites, this is the one they're going to try and push you to take. Been around for 50 odd years, commercial use. We could probably update that, make that 50 to 60 years now. So they are very widely used, but notice the size of the mite, of the predator. The top photograph shows an adult female spider mite being attacked by a larval stage of the predator. Look at the bottom picture, shows an adult female with the egg that she has just laid in front of her, and behind her you can see the smaller spherical egg of the spider mite. The predator egg is about 2.5 times bigger than the spider mite egg. They are also elliptical in shape, whereas the spider mite egg is perfectly spherical. So there is a complete difference. Once you've got your eye in on it, you can identify which is which. And when they come to hatching, it is literally the egg just kind of sprouts legs and gets up and walks away. So they are very useful predators. The other thing I would say is that this particular mite is what they call an obligate predator. It can only feed on spider mites, ideally two-spot spider mites, but they will feed on their own eggs and their own young if there's no spider mites around. So it is a curative rather than a preventative spider mite. those long legs allow it to get into spider mite webbing, and that's important for later on. Okay, next predator, Amblyseus andersoni. Notice that pest ratio is not as voracious as the Phytosulus. The photograph clearly shows the Amblyseid mite, and above it is their food mite. So these particular predators are not bred on spider mites, they're bred on what's called a factitious host. In this case, it's a flower mite that feeds on bran and other flower type materials to produce these mites. Also, this is licensed, or sorry, there is no license requirements for this predator in the UK. Very useful predator, and thus the bottom line of it is half the cost or less than of Californicus. So Andersoni, I'm not going to read through what it does, but the bottom line is the TSSM is 2 spotted spider mite, fruit tree spider mite, citrus spider mite. It also, we know it does Eotetranicus, it does thrips. Okay, not wonderful on thrips. I wouldn't use it as a frontline thrips control. Rust, gall mites, tarsnipid mites, the whole lot. So it's a very useful predator to introduce as more of a preventative than a curative. Okay, Californicus is the one I think most people would have heard of. Again, that pest ratio is not as voracious as Phytosulus. They're roughly the same size, maybe slightly larger than the predator, than the prey, but licensed for protected crops in permanent structures. This is a glass house. So again, it has license restrictions in the UK. The name Californicus would indicate where it comes from. So it is tolerant to high temperatures as well as low temperatures, but more importantly, it would more tolerate to low humidity. So this is a very useful predator if you can tick the right boxes for the license requirements. Just want to draw your attention to the bottom line of this table. You can see just how efficient the Phytosulus is on how much prey it will take compared to the Amblyseid mites. So we're talking about nearly 3 times more efficiency, more prey eaten by Phytosulus. But at 35 centigrade or above, Phytosulus will cease to feed. They will go and hide lower in the canopy. When it cools down, they become more active again. So there is a cutoff as to which predator to go for under which conditions. This case, I want you to look at the second line up from the bottom, third line down from the top. Just look at what happened when you drop the humidity with Phytosulus. So 27 centigrade, but at 80% humidity, fantastic. You're getting really good reproduction. You're getting eggs being laid, hatching, reaching adulthood. This is brilliant. They will actually knock out spider mites completely. Drop that humidity and it starts to go completely wrong. So again, Phytosulus, brilliant predator, but under the right conditions it will work. Get the humidity wrong, high temperatures, forget it. It's not going to work. They will go and hide in the lower canopy of the crop. So, which predator to use? So again, I've put some crosses on the table there. So Phytosulus, brilliant on predators, no good under low humidity, no good without the prey, and no good on alternative food sources. Whereas both the Amblyseid mites can be put in as a preventative treatment rather than a curative treatment. High temperature, if you can get the right license, I would say Californicus is the better one. Andersoni is very good on all conditions, indoors, outdoors, low temperature, so you can put it in early in the season as a preventative. So honestly, if you had spider mites on plants or in a crop last year, you're very likely to get spider mites in the same situation, same position in the greenhouse, might not be the same crop, but the same position because they'll overwinter off the plants. That's where Andersoni would come in as extremely useful introduction in late winter, early spring. Seems like I'm pushing Andersoni. I'm not. I just want to get this one up to date on things. It's a very useful predator. There's the range of crops it will feed on. So fungal spores, plant sap, it will survive in the absence of prey. Again, not pushing this one, but it would take it's a very useful predator. Anyway, I'm not going to harp on about Andersonite. It's very useful. Last predator to introduce is Feltiella acarasuja. This is a very useful predator. It's again an obligate predator, feeds only on spider mites. The adult is a winged fly, it's a midge. So again, host location is very, very good, but it needs a fairly high number of spider mites before they cut in to do the job. It produces eggs in amongst the spider mite colonies. The eggs turn into a small orange larva and they bite into the spider mite anywhere on the body and suck it dry, wriggle around and onto the next spider mite. So where you have colonies of spider mites, this is a very useful predator. Once they've fully developed, they will slide up to a leaf vein, spin a silken pad and pupate within that silken cocoon. So again, very wide host range. It will take out other spider mite species as opposed to just Tetranicus. This will do Panonicus, will do the whole lot. So external, indoors, outdoors, protected, non-protected, any situation, Feltiella is around. It's a very useful predator. They're not cheap, but they are very useful. Okay, looking at the pesticides, I've had to make some notes on this because it gets a bit more complicated. Looking at Apollo, it's what's called an overside. An overside will kill just the eggs, so no effect on adults, no effect on larval pupal stages or anything like that. It's purely an overside to kill the eggs. This one is a magenta pink color and it has an approval for outdoor ornamentals only. That's under an EMU, an off-label approval. Borneo, contact translaminous that moves through the plant. This will kill eggs. Look at the IRAC code numbers, 10A, 10B, so they're very closely related. Borneo will also make the adult female sterile. So it prevents any further egg laying, but doesn't kill the adults. It has effect on the molting stage. So spider mites, as they molt, it can kill those out. It has an off-label approval for protected crops or protected ornamentals. Dynamac, contact translaminar, kills the mites, doesn't do the eggs. This one has a protection, has an on-label approval for permanent protection full enclosure. It lost outdoor crops a year or so ago. Floramite, again, I'm not Flipper, Majestic, they're fine, IRAC exempt. Floramite is permanent protection on label. Naturalis is on label. Nealta is a new acaricide, different code number again, same code number as Floramite, related, so don't go using one after the other. That one again has ornamentals on label. Sprucet again, contact on label. Tech Bomb, contact, but we're still awaiting an approval on ornamentals. It has protected edible crops. At the moment, we don't have protected ornamentals. That will come. Okay, running through rapidly, looking at thrips, same layout. So we're going to look at the different pest species, the biologicals, and then wrap up with the chemicals. So Western flower thrips, WFT, it was originally a notifiable pest. This is back in 1986. It's now the most common spider mite species that we have on any crop, indoors or outdoors. The more important thing about this one is that it feeds on pollen and causes premature senescence of the flowers, but also causes that streaking on the flower itself. So visible damage to the flower plus premature early senescence of the flower. So reduced shelf life of the flowers themselves. But the bottom line on this one is virus transmission. This particular thrips will transmit several plant viruses. The photograph shows Alströmera and Astra fricatii, and this is the spotted wilt virus. And on the Astra, it was identified by plant health. So on a nursery at the same time, Andrew took some samples away, got them identified, and it came back as spotted wilt virus. So that's the sort of damage that we're looking at. So it's not necessarily going to wilt, but it will have those yellow patches on the leaf, and that's spotted wilt virus. Okay, very quickly on the life cycle, I think most people are aware of it. eggs are laid into the plant tissue. That's virtually anywhere. So it could be a leaf, it could be a stem, it could be the petal, it could be the anther. Anywhere on the plant, it will lay an egg into it. So that's inserted in the egg, in the plant. So it's protected. That egg will hatch to a 6-legged larva, which will feed on the plant, has a molting stage, goes to a second instar larva. At that stage, it does more plant damage. So they feed dramatically on the plant, doing a lot of physical damage. They then go through a pre-pupa and pupal stage. Now that can be on the plant. If it's a very large leaf plant, it can land on the upper surface of a lower leaf. If not, they will drop to the ground and pupate in the ground itself. I did trials back in the 80s, early 90s, and they go to a depth of about 5 centimeters in the ground itself, in the compost. So they will stay in there, they can overwinter in there. So it's important to understand where these thrips are actually laying in the ground. Size-wise, it's on there about 1.5 millimeters for an adult female. That's large for thrips. Okay, next one, the JFT, Japanese flower thrips. A new species, different, not the same genus. The Western flower thrips is Franklinella. This is now thrips itself. These are new to the UK. They've been in Europe. It's not notifiable. They're already fairly widespread. But this particular thrips, although it's classed as a flower thrips, does not feed on pollen, but will damage the flowers. But mostly it feeds on the leaf itself. So the photograph in the center shows a hydrangea leaf, and you can see the damage done by just a few thrips on those plants. Just for comparison, because it's a darker black thrips, is a cereal thrips, and these will only feed on grasses and cereals. So again, the thrips setosus doesn't pupate necessarily in the compost, but it is certainly associated with the compost. So if you're buying in hydrangea as a bare plant with a bit of compost, you could likely to get thrips setosus coming in with those plants. That seems to be the way they come in. So just a little bit on thrips setosus. It's polyphagous. Remember the broad mite, Polyphagotarsinemus. This is polyphagous. So polyphagous feeds on a whole range of different plants, very similar to Western flower thrips. The color is different, it's much darker, but it has that orange markings on the wings. They're quite characteristic. Virus transmission, the same as spotted wilt virus, or sorry, just spotted wilt virus recorded, whereas the Western flower thrips is up to 5 different viruses. Similar life cycle, so eggs, larvae, pupa in this compost or off the plant. So again, important to understand what this one will do. Other thrips to concentrate on, we have the echinothrips. I never guess where this one comes from. There's a bit of a clue in the name. So another import came across from plant or from America. This is what's called a gregarious species. The previous 2 were territorial. They will disperse very readily. These do just the opposite. They live in colonies together on the same plant. Heliothrips is the common name, is the glasshouse thrips. That is also becoming more common, particularly on protected crops. Notice the photograph, not that clear, I'm afraid, but it shows little droplets of liquid on the thrips themselves at the rear end. That is liquid frass. They use that as a defense against predator attack. So they can dollop that liquid frass over the predator, which puts them off. So again, useful to understand how this one works. This is a close-up of these gregarious thrips species. Photograph on the left has 10 adult thrips along that central vein. You don't see that with the other thrip species. Photograph on the far right shows a whole colony of the heliothrips on a leaf. But notice how they've grazed one section of the leaf. They haven't gone across the ridge to the second section of leaf. So the damage that they do looks completely different. In fact, if anything, it looks more like spider mite damage from above. But you'll see these whole groups of thrips together on the plants. Okay, so again, identification is quite important. AHDB have done a very, very useful poster, and that's a downloadable poster and it has various thrips species, colour photographs of what they look like, the adults, the larvae, and the damage that they do. Because we seem to be getting a lot of thrips from abroad, I've managed to find this on the internet. It's a field identification guide to thrips. Very useful, but don't knock it because they're just American. That seems to be where a lot of thrips are coming from. So again, the AHDB poster is well worth downloading. In the previous presentation, I talked about sticky traps and how they work. This is something that you can add to improve the use of sticky traps against thrips. So the top one is from Bioline. It's called Thripline AMS, and that's Advanced Monitoring System. And this is what's known as a sex aggregation pheromone. So an aggregation or sex pheromone attracts generally males to the scent of a female. In this particular case, it's an aggregation pheromone, and it works on both males and females. So the photograph of the blue sticky trap shows the pheromone at the bottom end of the sticky trap. Now, if it was just the pheromone working, there'd be a concentration of thrips around the pheromone and not evenly spread over the entire blue sticky trap. So what tends to happen with these, it draws the thrips out of the flower or out of their hiding places. The colour takes over to attract the thrips to them. So you can use it as an advanced monitoring to extract Western flower thrips. It only works on Western flower thrips, it's quite specific. Or you put the pheromone out a few hours before you make a pesticide spray. That again improves the efficacy, particularly for contact sprays. You get the thrips out and you get more chance of hitting them. The bottom two are both chiromones. Those are slightly different. A chiromone is the scent produced by something which has no benefit to the producer as such, but it does attract the pest to go to where that scent has come from. So it's there to attract, and it improves the quality of the sticky traps by about 3 to 4 times. So it does dramatically improve the efficacy of sticky traps. Now, which to go for, yellow or blue? You can use yellow sticky traps and they will catch pretty much as many sticky thrips as a blue sticky trap, but a yellow trap will also attract other insects. So we're looking at adult whitefly, adult aphids, and so on and so on. So if you're trying to control thrips and you've got aphids and other insects stuck onto it, leafhoppers, big insects and moth flies, you're losing a lot of potential sticky area. So there is some scientific papers. Claire Sampson's done a very, very useful PhD on the use of sticky traps. But why not use both? This is a trial that was done with blue roller traps and yellow roller traps, and the contrasting sticky trap plastered onto it. And the photograph on the right-hand side, commercial nursery, yellow sticky traps with strips of blue stuck on it. And in this case, we have what's known as the edge effect. We tend to get more thrips around the edge of the yellow and the blue, or the blue and the yellow, where the two meet on the trap. Again, that tends to pull out more adult thrips, but less of other insects on blue. So it just makes the blue sticky traps more efficient. Okay, looking at control, we have the Amblysseiid species. Again, some of these are now called Neosulus, some are Typhlodromus, some are Typhlodromips. So just to be on the easy side, let's just call them Amblysseids because they are Amblysseiid mites. The Amblysseiid mites tend to attack just the first instar. Some will have a go at the second instar larva, but it's generally the first instar larva of a thrips. So it's not much good as a curative, although they will work, they will help. It's more of a preventative for thrips control. So you put the predators out before you start to see thrips, or you suspect thrips are going to come in. Other advantages of the Amblysseiid mites, they will feed on pollen, trichomes, glandular hairs, extrafloral nectaries, the whole range. So they will actually be there before the thrips starts to get going. Again, I've just picked on a couple of slides. They will work in sachets, and the sachets are waterproof, so you can get a reasonable amount of water onto them, providing it doesn't go into the hole which is punched on the other side. The Gemini sachets, which hang over a plant, brilliant because they would do the gall mites, they would do the whole range of different mites, indoors or outdoors. On with predatory mites, we have what was known as Hypoaspis miles is now Stratiolaelaps. Again, same mite, just a different name to the mites. I believe some of the companies still refer to it as Hyposure, Hypoline, etc. So they still refer to the old trade name as the hypo. Usually introduced to control sciarids in the compost, sciarid larvae, but will also give a good degree of other pests that they come across. So we're talking about thrips larvae as they drop to pupate, thrips pupae, symphalids, springtails, etc. So useful background predator to introduce to each batch of plants. The aureus, this is what's called a true bug. So it has a stabbing proboscis, it stabs into the prey, generally lifts it off the leaf so its little legs can't run away, sucks it dry and on to the next one. We did experiments about 25, 30 years ago where we looked at aureus and we're up to 30 odd thrips per day being eaten by an adult aureus. Their nymphs are also very predatory as well. So if possible, a source of pollen, because they do like a source of pollen. Again, work's been done. Jude Bennison did some work looking at introducing Alyssium into strawberry crops. Okay, we're looking at ornamentals today, but the general inference is there. If you have a source of pollen somewhere along the line, aureus are going to be a very good predator for the adults and second instar larvae. Lacewing larva, again, generalist predator. Photograph there to show it feeding on a second instar thrips larva. Yes, they will feed on thrips, but it's not going to be the first line of defense against thrips. It's just useful to have lacewings around the place. Pathogens, so we have the Bovaria and Lecanicilium. These are commercially available, so we have Botanigard, Naturalis, Mycotal, fungal pathogens, but they are all temperature and humidity dependent, critical. Nematodes, I've put those under pathogens because they are classed as entomopathogenic nematodes because they carry the bacteria with just the killing. These are applied as a foliar spray and they will work very well on flowers where the flower is pointing upwards to get the nematode, the spray, into the flower. If you have flowers like cyclamen, which is pointing more sideways, not so good. If you're on crops like fuchsia, where the flower points down, forget it. But these will also feed on the thrips pupae, so the late larval stage and pupae. So even if you're on the wrong sort of flower, it's worth looking at nematodes if you have a major problem, a line to actually get it into the compost to actually knock out that larval stage, pupal stage. Okay, looking at pesticides. Now I need to go back to my notes to find out which is which. So azotin, on label approval for ornamentals under full and permanent enclosure. Batavia as an off label approval for protected ornamentals. I'm not going to go through the others because I've already done that. Conserve on label, Dynamec on label for protected. The Flipper Majestic, Plant Invigorator, Protac, again, they have the approvals. Gazelle has outdoor and protect ornamentals on label. Mycotal, Contact, protected ornamentals on label. Spruce it, again, protected ornamentals on label. We have looked at cocktails. I have found that Gazelle, Dynamec, and a product called Atraca work very well in combination. Gazelle, as I say, Gazelle Dynamec, Gazelle Conserve Atraca is also a very useful cocktail to go for. This is my final slide on Atraca. It's a copper product and it is liquid sugars, so fructose, glucose, sucrose. It's what's classed as an appetant. So it improves the appetite of the pest to feed on the spray deposit. So if you mix Atraca with any pesticide aimed at thrips, spider mites, caterpillars, sawflies, vine weevils, you will improve the efficacy of your product. So Atraca is a very useful addition into the armory to control those particular pests, but you add it to any pesticide aimed at those pests. No benefit to adding it to aphid control or other things like that, or leafhopper or whitefly, but certainly for pests such as these, a great benefit. Okay, that was my presentation. I think we're now back to Wayne and on to James. Thank you.
Thank you, Neil. Just to remind you all, if you do have questions, please fill in the box and send them through, and I'll keep an eye on them and pose them after James's presentation. But as Neil says, if we can move on to James's presentation now, and we're talking about Thrips setosus specifically, and the way James has managed to sort of get to grips with this particular pest. So over to James. Thank you.
Hello, so thank you, Neil. That was very interesting. Now I'm going to give a slightly different perspective. I'm going to talk about my experience of thrips on our nursery. I'm going to be focusing on Thrips setosus, and I'm going to try to explain what they are very briefly, where I see them, when I see them, what they do, and how I try to control them. This is all from my perspective as a grower. I'm not a researcher or an entomologist, but I've had to learn this by necessity. So, Thrips setosus, it's a fairly recent pest. It's been in Europe for 6 or 7 years, and I think it's been in the UK for about 5 years now. We had our first confirmed case in 2019 on our nursery. I have a feeling we may have had it the year before that, but the first confirmed one was in 2019. So I'm going to compare Thrips setosus to Western flower thrips because up until recently, if you said thrips to me, I would have immediately thought Western flower thrips. It was the main species which I had to deal with. But setosus is different to Western flower thrip in a number of ways. Most obviously is their appearance. The adults are black with a sort of pale band around the top of their wings. And the larvae, I can't really tell the difference. So Thrips larvae look like Thrips larvae to me. Their appearance makes them easy to see. To find them, I tend to look on the underside of the leaves because that's where they seem to be most of the time. And that's for both adults and larvae. If I was looking for Western flower thrips, I used to go around and tap the flowers, patch them out onto a piece of paper, and then I would see the thrips there. Whereas the setosus spend almost all the time on the underside of the leaves, and they seem to do a lot of damage to the leaves as well. Another difference between them is the way that they need to be controlled. Some controls work for both, not all are true. So they're both thrips, but they are different and they do need to be treated differently. So where do I see Western flower thrips? I've seen them initially on cyclamen and hydrangeas. I've seen them on most flowering bedding plants. I've seen them on plants which we've brought in for the nursery to trade in or the garden centre. I've seen them on even big foliage plants, orchids, and interestingly, I've seen them on final stage poinsettias and appear to have all stages of the life cycle on there. So it started off for us as a seasonal problem. Usually in the autumn, I'd see the setosus on cyclamen. On the slides, you can see some damage which has been done to the flowers, but the main damage is to the underside of the leaves. They will completely destroy the undersides of the leaves, which then causes other problems, leads to infections, botrytis, and obviously they can transmit virus as well, as Neil mentioned. The problem so we had a biological control system in place. We had methods of controlling Western flower thrips, but when we used those same controls on setosus, it didn't appear to work. So I would end up using the same controls. I would try to increase the rates. I would put more cucumis on there, which was the main sort of preventive. Would be using cucumis and hypoaspis, and it didn't seem to be working. So increase the rates, that didn't work. So what else could we do? I'd ask advisors and see what they'd say. I'd get optimistic and think that perhaps there's something new on the market. Maybe it was azotin. So I would try that. It may well have had an effect, but it wasn't the cure that I was looking for. It did not fix the problem. I tried Aureus as a curative, but that didn't have an effect quick enough. The problem still existed. Sometimes I carry on for too long with the biologicals. This would lead to poor plant quality, and we would have to waste the plants. This cost us money, a lot of time, more products. It was not a good situation to be in. And the frustrating thing is I didn't seem to have any answers at this point. I would ask plant health to come and have a look, and they would confirm that, yes, this is setosus. It's not regulated, so don't worry. Have you tried Dynamic? Yes, I have, but that didn't do the job. So I found that Desis Forte worked. I did a trial once and it worked very well, and I was amazed. So I used that as my sort of get out of jail card. But I needed to fix this. We had a loss of confidence with the biological controls, and this was not great because then I'm asking staff to apply the biologicals, and they're not believing that they work. They don't believe they work. So I was determined to find a problem. I was determined to find a way to break this cycle. I needed a sustainable solution. So that was last year, and then this is February 2021. So we grow a lot of New Guinea impatiens. We pop them every week from January through to June. It is our single biggest crop. So I mentioned I was determined to fix this problem. In February, when I sort of hoped that these little things might be in their diapause stage, there was no diapause going on. They were very busy, prolifically breeding and munching on my New Guinea impatiens. They were doing a huge amount of damage at a time when I hadn't even started the bio program. I did not expect them to be this active so soon. Felt like it was the middle of winter. It was dull. It was not a time which I expected thrips to be flourishing. Now, in the past, I had used Desis to nuke them, and that was my sort of, as I mentioned, get out of jail thing. But this is the start of the season. I knew that I could not start using Desis now. Firstly, it was becoming less effective, even though I had tried to use it very sparingly. I'm sure resistance was building quite quickly. So I needed to look for a proper solution. So I spoke to everyone I could. I spoke to entomologists, I spoke to researchers, crop protection suppliers, young plant suppliers, Dutch growers. But it was clear there was not a simple solution. It wasn't a case of spray with this, it will all be okay. So I had a problem and I had to fix this because this could not carry on all the way through the year if I couldn't control it in February. So I started experimenting. I looked at various insecticide catalogues. I read through every product label. I did spray trials. I was testing the activity of different active ingredients and also testing for phytotoxicity. And the interesting thing I found was that sometimes I would get different results from doing a trial to an actual real life spray on an entire crop. I think one of the reasons for that is that doing a trial, be with a knapsack, I can make sure I've got excellent contact, could spray all over on the underside of the leaves. Whereas in reality, when you're spraying a big area, it's not possible to spray the underside of every leaf. So to try to help achieve contact, we used high pressure, we used silicone wetters. Now, the other interesting thing was I found that using a sort of a tank mix or a cocktail appeared to be more effective, some products, than using those same products on separate applications. Up until now, we'd used tank mixes in the past, but I always thought that it was just a convenient way to apply multiple products in one go, but I hadn't realized that the effect of them all being applied together can actually be it's more powerful than applying them individually. So we used a number of products and we got the population down, which was great. I was very concerned because it could have been a business critical situation. We had to find a solution, and now seemed like a long time. It was probably fairly quickly. We had reduced the population, certainly not eradicated it, but it was down to a more controlled level, so we could start looking at introducing some biological control agents. So the main one we used was Montdorensis. It proved to be very good. I started off quite high rates, going considerably over 100 per meter squared, and then gradually reduced it down as my confidence grew that we had control and I was not seeing any more Thrips larvae. So then I reduced it down, and I think we sort of settled to about 30 per meter squared now. I also started introducing feeder mites as well, and we would mix them in with the Montdorensis and apply them at the same time. This I think helped to keep them, or helps for the population to establish. Some people say that you should not put Montdorensis onto very young plants if there's no pollen, or I believe for them to feed on, but I've been putting them on all the time, and I can see that they are surviving. They're there. I did use some Aureus at one point, and I also used some nematodes and Hypaspis for control of the pupae. However, I stopped using these products because I felt we had control with the Montdorensis, and as I was not seeing the larvae, therefore I didn't see the need to be controlling the next stage in the life cycle. So this is our IPM plan, starting at the top with the Amblyceus Montdorensis mixed with the feeder mites. On the left-hand side of this slide, you can see our technical way of mixing these two, which is a bucket. And then on the right-hand side, you can see us, or not us, this one, you can see the copper mini air bug, which has been good. It's been very helpful for applying the bugs, because it's all very well when you read the manuals and they say, oh, you should apply them at X per square meter, but actually trying to achieve that is often very difficult. So this appears to have worked. I can see them. They do exist. They're living things on the underside of the leaves. So starting at the top, I've got the Amblyceus Montdorensis, and my plan I assumed that the population would increase again, and I would then have to increase the rates of the Montrensis. And then the next stage on from that was to introduce some Aureus. Nice thing with Aureus is apparently they should target all stages of the life cycle. I used them at the very beginning when we were just still trying to mop up some of the initial population, but I have not had to go back there. And the same, we have not so far, touching wood as I say this, we've not had to go back and do any chemical applications. But the weather this year has been slightly atypical, and I'm under no illusion that we've found some miracle cure and the problem is solved. So that's the plan. We go round in that circle. Ultimately, if we apply the chemicals, understand that depending on what we apply, we're likely to kill off all the beneficials, but that's just the way it has to be. I think there's times when you just have to accept quickly take the action to move on and minimize the crop damage. But so far we've stayed at the top of this cycle, which I'm very happy about. In fact, last week I saw 2 adult Thrips setosus on a weed, and I've not seen any this week and haven't seen any in previous weeks to that. So it gives an idea, whereas before that I could have found many thrips on one leaf of a single plant. So what have I learned from this? Well, in fact, now I'm going to go back slightly. I wanted to, at the start of this presentation, I said I want to cover the thrips, where I find them, and the answer is potentially on everything that we grow, almost. When do I see them? Well, all year. What do? They clearly destroy our crops. And can I control them? Yes, so far I think we can. It's early days, but I have a lot more confidence now, and our staff have a lot more confidence. So what have I learned? I've learned that the biological controls can work. It does take a lot of discipline and commitment and planning. And when I was reflecting whilst writing this presentation, I realized that it's very easy to see when the controls don't work. But when they do work, it's easy to forget about that and to not appreciate it. So it's good to know, or to remember that actually they are doing their thing in the background. If you don't have a problem, it might just be because they're doing their job. I've also made a point of doing my own biological plan rather than having someone else, a supplier, doing it for me. That's helped a lot, because it's all very well having a potting plan and then a biological control plan to go with that. But sometimes on a nursery, things don't happen exactly to schedule. Things don't get spaced or potted exactly on the week that you say they should. So it's useful to be able to adjust the plan accordingly, and also to really understand what the products are doing, to then be able to adjust them as and when needed. I've also learned that scouting pays off. I've been picking up on other pests a bit sooner. This year we've had very few problems with spider mites. I'm sure that could change with the recent hot weather, but I also think that the Montrensis have probably been helping. When I have spotted spider mites, there's often been the Montrensis nearby, and because I've invested heavily in the Montrensis, I'm reluctant to go in and spray with Dynamec or something unless I have to. So I'm more inclined to use some phytocidus or something to help mop up the odd little bits of spider mite. So nearly done now. Other things I've learned, to my surprise, that we used big roller sticky traps at times. I was amazed when I put those big roller traps out and then put some small monitoring traps in that same crop. the monitoring traps, although a far smaller surface area, they were actually catching more adult setosus than the big roller traps. And the other thing which I wanted to mention, which I haven't so far, was that one complication when talking to my Dutch colleagues is that they have access to different active ingredients. So they use a product called Mainspring, which is biocompatible. I tried to get hold of that, and it turns out, yes, that it is approved for use in the UK, but it's not. There are restrictions which make it unusable for us on our nursery, and so it would be really helpful if there was if we did have access to this product in particular. And that is it. So I'd just like to say thank you very much. If anyone has any experience with dealing with setosus or you have different views to me, I'd be really interested to talk about it. Feel free to email me or ask some questions now. The same if you've got questions for Neil, please fire away. Thank you very much.
Thank you, James. Thank you, Neil. If I can start with Neil, coming back to the mites. Once the mites are going into hibernation, and in the case of the spider mite changing colour, how susceptible are they to cleanup sprays at that point? What actions can you take?
Providing the spider mites are still on the plant, then most acaricides will work. But once they've left the plant, you have no access, no contact with those mites. So it is absolutely critical late September, by mid-October, to knock as many spider mites off as possible.
Okay, thank you for that. And just picking up on the broad and gall mites, you did mention 1 or 2 biocontrols for them, but is there any specific recommendations for the control of the really small broad gall mites which tend to get into the growing point?
Yeah, I mean, basically any of the amblyssaiid mites are very good. Even cucumerus is very useful on proper broad mite and tarsnibid mite. So if you're a cyclamen grower and you're introducing cucumerus for western flower thrips, you very, very rarely get much damage from tarsnibid mite on cyclamen crops. And that's purely down to the amblyssaius nearsius cucumerus, the same thing. Okay,
thank you. I'm picking up on your last slide. We've had a question about, is there a recommended rate for a tracker?
Yes, there is. You want to know what it is now?
Yes, please. So I should have asked an open question rather than a closed question, really, shouldn't I? Yeah, and is there a rate, and what is that rate?
Right.
Neil,
yes. The tracker rate is between 1% and 2%. Or is it 0.12%? It's 1 to 2 mil per litre, basically.
Okay.
1 to 2 mil per litre. 2 mil per litre for the bigger insects. Generally 2 mil per litre for most things. It does not make anything sticky. It's fine.
Okay. And coming on to James, Neil mentioned the fact that you can spread tomato spotted wilt virus. Did you have any instances on any of the crops with tomato spotted wilt virus at Cobbins as a result of setosus?
Not that I'm aware of, no.
No, okay. And do you think now you're having to deal with a resident population year on year, or is it still a case that you're thinking you're buying it in still, and it's a bought-in issue with certain crops you're buying in?
No, I assume that we now have a resident population on our nursery. Having said that, I'm sure that one particular crop they probably do come in on.
Okay.
Yes.
And we've had a question about the feeder mites. Is it a product name for the feeder mites? Is it a specific mite? Perhaps Neil may be able to help here as well.
Yes, James has been using the one from Bioline, and it's just called Mite Food.
Okay.
James, you want to say anything more on Mite Food?
No, that's fine. We mixed them in, and yes. There's not much more I can add to that.
I think it's a bag of 10,000 mites. This is a prey mite, but it's a live mite for the amblyssaius and the other insects to feed on.
Okay, thank you, Neil. And you mentioned trapping and using small yellow traps to monitor for setosus, James. With it being not particularly a mobile mite, are you finding there's more emphasis on scouting to keep abreast of populations rather than using traps?
The traps are useful, but I do find that if there's a population, they're quite an easy one to find. It's easy to find them through scouting.
Okay. And regarding thrip influxes, just coming back to Neil now, quite often in the summer you do get influxes of the cereal thrips, and you mentioned before they don't feed on well, continue to feed on crops, but they may try a crop out and impact on bio control programs. Is there any tips in terms of managing thrip influxes coming in through the vent at cereal harvesting time?
I think it's the old Dad's Army phrase, don't panic. They tend to come in, they will feed, they will do a little bit of damage, but they will not last very long on non-cereal type crops. The worst thing we see is on poinsettia, because it's obviously started off in July, and we're going right through the harvest period when cereal thrips are around. If you're in the wrong direction, they will come in, they will land on the plant, they will do a little bit of damage. It's basically 100% waste of time putting cucumis or any other predator in there to control it, because they won't like that plant, they can't reproduce on it, and they'll be up and off.
Okay, thank you for that. And then one final question then to Neil, so we can sort of wrap up, because it's getting towards the end of today's webinar. You mentioned that setosus isn't notifiable. What about the other thrips you mentioned in terms of the Thrips and Helyer thrips? Are they notifiable thrip species?
No longer, they're just here, I'm afraid. Certainly the glasshouse thrips has been around before Western flower thrips.
Okay.
So what tended to happen when Western flower thrips came in, what was aimed at controlling that was automatically controlling the glasshouse thrips, the heliothrips. Now we're looking at more biocontrols. We're looking at heliothrips not becoming a major pest, but it is more evident at the moment.
Okay, thank you for that. Right then, if I can just wrap things up by thanking both Neil and James for today's presentations. Thank you to both of you for quite quite extensive presentations, and also for giving us a really useful insight into setosus, which I think for many people is a relatively new pest. If you haven't downloaded the handout yet, you've got a few seconds just to download the handout before we bring the presentation to a close. If you do have any further questions you've not thought of in the next day or so, please send them to me, and I'll pass them on to Neil or James as appropriate. As I said, the recording will be made available next week, along with the first webinar on aphid and whitefly control. There is a AHDB Thrips review, which was undertaken by Drew Bennett and ADAS. If you receive the monthly update today in your email box, there is a link to that and a few extra notes about thripsitosis. So if you want to brush up a little bit more on thripsitosis, there is a complete review there, summarising some of the Dutch work that's been undertaken for the last couple of years. So with that, I say thank you to both presenters, and I wish everybody a successful end to the current bedding season and future success for the remainder of the year. Thank you for joining us.
Thank you. Bye-bye.
Thanks. Bye.